if you live in countries which are TB-free, not really, right? But it's much more controlled. There are certain, immigrant populations to be tested and so on, right? And if you present with certain, presentations, then you might get assessed with it's quantiferum. So you get basically exposed to the peptide, right, the protein, and then see if you get a T-cell response, a recall. Yeah. So you can detect like a recent infection or some kind of exposure in the past. Yeah. That's how you test it. You get chest X-ray as well if you're appearance positive, right? But no, it's not prevalent in this country anymore. But in other countries, yes. Yeah. And so they get BCGs at a young age. Not really protective, but that's normal. No, treatment is antibiotics, right? And in those countries, it's not good. It's not well managed. Yeah, so that's why you get these hyper-resistant, multi-drug resistant XXX variant forms. Yeah, because you just go buy some antibiotics from the market, right? And then so they drive emergence. So many countries very.
Okay, so good morning, everybody. Thank you for coming along to the lecture. It's actually really good to see so many of you, especially at the end of the semester. It would be great, actually, if our neuro students did the same. Today I'm actually going to continue on probably from what Jamie's kind of started in your neuropharmacology series. I'm going to provide a real whirlwind tour, really, of two kind of major drug classes, which is cannabinoids and psychedelics. And normally, actually, in neuro, I usually give 2 lectures on cannabinoids. So today is really going to be whirlwind, but it's really just to give you the fundamentals of what these classes of drugs do, what they are, and how they actually change the nervous system, and then a little bit about how we can use drugs to maybe target these different types of systems. So we'll start off with endocannabinoids or cannabinoids. So in the literature, you might see things like cannabinoids, endocannabinoids, phytocannabinoids. So it's all about cannabinoid system. When we talk about endocannabinoids or the ECS, we're talking about cannabinoids that are actually made endogenously. So that's where the E comes from. And so endogenously, the endocannabinoid system, it's really important actually for neural development. So, from all the way from like embryos to like early postnatal development, endocannabinoids play a really important role. But they're also expressed in the adult nervous system, and they're kind of the main role in the adult nervous system is to modulate neural activity. So one of the, we'll get to it, but one of the main takeaways is that endocannabinoids are really regulating neurotransmitter release. And by regulating neurotransmitter release, they're really regulating neural activity, which is important for things like general function, but also things like neural plasticity, which I think you learned a little bit about in PHAR3320. So the endocannabinoid system is kind of like a lot of the other systems you heard about in terms of some of the neurotransmitters. So we have receptors, we have things that make them, things that move them, things that break them down. So in terms of the endocannabinoid system, we have cannabinoid receptors. And so some of you will be familiar with this diagram before. So this is showing the presynaptic neuron, this is showing the postsynaptic neuron here. And straight away you'll notice that the endocannabinoid receptors in this diagram are only being shown on the presynaptic neuron. And so what's not shown here are glial cells, but in terms of neurons, cannabinoid receptors are only expressed really on the presynaptic side. So we have cannabinoid receptors, which we'll talk about in more detail. We have the cannabinoids themselves, the actually endogenous cannabinoids, which we can see here, 2-AG and anandamide. We have proteins that actually make these cannabinoids, and actually so we have some enzymes here, and we have some transporters that move the cannabinoids across from the postsynaptic neuron to the presynaptic neuron. And then we have proteins that actually break these cannabinoids down. It's a very complex system. I'm not really going to be able to kind of convey that complexity to you today. But it's important to know that endocannabinoids really interact with other systems. And so and they interact with not just the brain, but also other systems like the bones. And that's why understanding cannabinoids can be a little bit difficult. Today I'm really only going to talk to you about how it affects the nervous system. So one thing to get across to you is that endocannabinoids, they're made and they're transported and they affect things like neurotransmitter release, but they themselves are not neurotransmitters. And so I think if you came to some of my very early lectures in PHAR3320, I showed you probably this diagram which was describing to you what is the definition of a neurotransmitter, right? So if we look at the first two things on my list, it said that neurotransmitters had to be synthesized in the presynaptic neuron, and that if it was present in the presynaptic neuron, it would be released in amounts that can cause a defined action on the postsynaptic neuron. And so straight away, this kind of indicates to us that this is why cannabinoids cannot be neurotransmitters, because the fact that they're actually not made in the presynaptic neuron, they're made in the postsynaptic neuron. And when they're released, it doesn't have like a defined action on the postsynaptic neuron, because again, it's having a defined action on the presynaptic neuron. And like I said, it does regulate things like glia, but we won't talk about that. So, but the thing is, it does do the other two things. So, if it's released in a reasonable concentration, it kind of mimics what some neurotransmitters can do. And there is a specific mechanism to actually remove it from the synapse, and that's a transporter, which we'll talk about. But really, the bottom line from this thing is that endocannabinoids, I guess, are somewhat similar to neurotransmitters, but they can't be because they don't show these first two properties. The other thing is to say is that I'm shown here is that I talked to you about in first semester about how neurotransmitters are released in vesicles. Endocannabinoids are actually polar, so they can actually move without the use of vesicles. So endocannabinoids don't get packaged and moved in vesicles as well. So what are these endocannabinoids? You're probably familiar with them. So in terms of endocannabinoids, the ones made in our bodies, we have the two main classes are these things called anandamide and 2-AG, 2-AG being 2-arachidonyl glycerol. And like I said, these endocannabinoids, they're actually made in the postsynaptic neuron, and they're released backwards, I guess, into the synapse. And so they go from postsynaptic neuron into the synapse to affect the presynaptic neuron. So it's kind of moving in reverse to a normal neurotransmitter. They're actually broken down quite quickly by enzymes located intracellularly. So there are some enzymes located in the postsynaptic neuron, which kind of break it down before it's released. but most of it is actually broken down once it's gone into the presynaptic neuron to have its effect. There are then enzymes in that presynaptic neuron to break it down, to stop it from having its action. So these are what the endocannabinoids, or endogenous cannabinoids, ECBs, look like. There are also other types of cannabinoids that we can make them, so they can be synthetic, but as I'll talk about, there are also plant-derived cannabinoids. And when we talk about plant-derived cannabinoids, these are your phytocannabinoids. So anytime you see phyto, that means plant. The two enzymes that actually break down the endocannabinoids are this thing here called fatty acid hydrolase, or PHAR. So PHAR actually breaks down both anandamide and 2-AG, but 2-AG is broken down by another one called monoacyl hydrolase as well. Also, if you have any questions, feel free to stop me as we go. Or if I'm going too fast, I see a lot of you writing notes, so if I'm going too fast, you can feel free to slow me down. Okay, so we'll talk about the endocannabinoids in a bit more detail. So like I said, the two main types are this 2-AG and anandamide. So here we have anandamide 2-AG, again, only in the postsynaptic neuron in terms of where they're made. They are actually synthesized in this postsynaptic neuron, and then they're released from postsynaptic going towards presynaptic neuron. You can see here that thing I told you about PHAR, which breaks down things like anandamide. The other one, that monoacylglycerol, that's located here in the presynaptic neuron. And essentially, what endocannabinoids are doing is that after they're released from the postsynaptic neuron to the presynaptic neuron, they're really trying to inhibit neurotransmission from the presynaptic neuron to the postsynaptic neuron. And they do this because they act on inhibitory GPCRs, which we'll talk about now. Endocannabinoids are not like neurotransmitters, where neurotransmitters are kind of like made and they're stored at the presynaptic site ready for release. Okay, so endocannabinoids are really interesting because they're actually made on demand. They're synthesized on demand. So usually, like when we have when they need to be released, or we have like changes in neural activity, that's the stimulus to say, okay, to the postsynaptic neuron, we need to make endocannabinoids. And so they're made on demand, and they're made on demand because the postsynaptic neuron actually has these precursors, which are these phospholipids that sit in the postsynaptic neuron, and then there are enzymes that actually then kind of make the cannabinoids. So here, for example, for anandamide, you can see the phospholipids here, which are needed for it. And then for 2-AG, they're actually made also from this glycophospholipid. But like I said, they're made on demand. So when they're needed, it pulls these lipids and then breaks them, and then these enzymes act to make them. And so I guess it's a little bit slower than neurotransmitters, which are always ready. There's a bit of delay in endocannabinoids because of the fact that they're only made after a stimulus, which is usually neural activity. The phospholipid, which actually makes both of them, is that glycophospholipid. You don't need to worry about this extra amine. Just know that both endocannabinoids are actually made from this precursor, glycophospholipid, and it's located postsynaptically, made on demand. So once we make the endocannabinoid, we obviously then need to move it, okay? And so its movement is not something super complex. It's done by a transporter. So like I said, endocannabinoids themselves are actually polar, so they don't actually need vesicles to move around the neuron. They can actually move by themselves. And so once they're made, they need to be transported. And they're transported by this thing called EMT, or the endocannabinoid membrane transporter. And its movement is very simple. It's kind of what you learned about in first semester. It's just concentration gradient driven, right? So when endocannabinoids are made on demand, we have a buildup of endocannabinoids. That concentration gradient is then used by this transporter to move them from the postsynaptic neuron to the synapse. There's obviously now a greater concentration of the cannabinoids in the synapse, and so the transporter on the presynaptic neuron then uses that concentration gradient to move them. Okay, so it's just a very simple transporter, and it's concentration gradient driven. Because the transport is done by this EMT, it's then probably no surprise to you then that the EMT will be like a target for like drug action, and we'll talk about that later on. So once we have cannabinoids made and then moved from postsynaptic to presynaptic neuron, what do they bind to? So there are actually two receptor types. There's CB1 and CB2. So CB1 is a GPCR. It's primarily coupled to an inhibitory G protein. And CB1 is really highly enriched at the presynaptic compartment. So when we get released, there's obviously a lot of receptors for it to bind to. When it binds to its GPCR, you get this activation of a very rapid decrease of cyclic AMP, right? And it does this by inhibiting adenyl cyclase activity. Hopefully that's not a surprise to you guys. You've heard of GPCRs before, right? Yep, cool. So once it binds, it's a very a rapid effect. That's why it causes inhibition very quickly. And once it activates its GPCR, the way that it inhibits neurotransmission is because it actually inhibits the activity of the voltage-gated ion channels that sit at the presynaptic nerve terminal. So you might remember in first semester I told, maybe I or Jamie told you that when you get the action potential travel down the axon to the presynaptic nerve terminal, it actually activates these voltage-gated ion channels, and a lot of these cause things like calcium to come in. And when we get that to come in, that's the stimulus to release neurotransmitter release. So if the endocannabinoids are preventing these voltage-gated ion channels from actually even opening or becoming active, there's actually no stimulus to allow the neurotransmitters to be released. And that's how it stops or inhibits neurotransmission. Is that kind of clear to everybody? Yeah. So it binds to the receptor, the GPCR, it causes that change in cyclic AMP and adenyl cyclase that then inhibits voltage-gated ion channels that prevents neurotransmitter release at the presynaptic nerve terminal itself. There's then CB2. CB2 is also a GPCR that's coupled to inhibitory G proteins. The difference is that CB2 is actually mostly, to what we know, expressed on the immune cells of the brain. They are expressed on neurons, but their main function in neurons and glia is to, we think, involved in the immune and inflammatory processes. And that's why you find them on things like astrocytes and microglia. Its activation is the same, leads to a rapid decrease in cyclic AMP that inhibits adenyl cyclase, and then you get that inhibition of voltage-gated ion channels. Now, what's not going to go into great detail is that endocannabinoids are a bit tricky because they have their receptors, but they also seem to bind to other receptors. So things like GABA-A, cannabinoids can actually bind directly to GABA-A. And so GABA-A, as you might remember from semester 1 and 2, can also be expressed postsynaptically. So that's just a little bit of snip information, but for most of your knowledge, you just need to know where CB1 and CB2 are located and kind of what they do to their main function on the nervous system. Any questions so far? Okay, so that's the endogenous ones. Those are the ones we actually make in our nervous system. What about the ones we get from plants, right? So, talking to undergraduates, probably the idea of phytocannabinoids is not something strange. So, in terms of phytocannabinoids, there are actually quite a few out there. So if we look at just the cannabis sativa plant, there are over actually 100 phytocannabinoids that can be isolated just from that one plant. Probably the most one that's abundant and the most well known is this thing called delta-9 tetrahydrocannabinoid, or THC, which I'm showing here. And it's a partial agonist for CB1 and CB2 receptors. And it's actually the most prominent psychoactive phytocannabinoid from the cannabis sativa plant. The other one that's probably most abundant in that plant is cannabidiol, or CBD, also a partial agonist for CB1 and CB2. But one of the main differences between THC and CBD is that cannabidiol actually seems to lack the kind of reward and reinforcement effects that THC has. And so because of that, CBD is often considered to have less abuse potential compared to, say, THC. But you can see actually their structures in a diagrammatical form is actually pretty similar. There are just some subtle changes to the bonds that they have. So a little bit more about THC. So THC is actually itself made. So when we have the phytocannabinoid, it's not actually made until you actually heat it. So it actually needs to be burned. And the reason why you need to burn it is because you need to get this decarboxylation off from its precursor to the acid form. Okay, so you actually need a lot of heat to convert it to the actual THC form. It's very diverse in what it actually can do to the nervous system. And so that's actually very tricky then to use it as a tool and as a treatment, because if we look at just some of the ones I've listed, so THC causes a wide range of cognitive changes, a wide range of psychoactive changes. But it can also do things like anti-inflammation. Antipyretic actually means like anti-itch in this case. Bronchodilation, we can use it to treat spasms, muscle relaxation, things like anxiety. But it also has some things like impaired memory. Depending on what purpose you're using it for, it might be a good or bad thing that you can induce things like immunosuppression. And like I said, its main action is through a partial agonism of the CB1 receptor, but it has a very high affinity for the CB1 receptor. If we contrast that now to, say, CBD, I'm going to start at the bottom here. CBD has a low affinity for CB receptors, so that's also one of the differences in terms of its mechanism. People consider it to be the non-toxic phytocannabinoid because it seems to have less detrimental effects. It also induces a wide range of effects. So we can use CBD for things like analgesia, to treat inflammation. It's a good anticonvulsant, anxiolytic, and also antipsychosis. Its interaction with CB receptors is very complex, and so I won't bore you with the details. But all I want to illustrate here is that there's a lot of work being done on like CBD and THC, for industry, but also for for research. But they're very tricky substances to study because we don't really understand how they're interacting with the receptors. I've just put here an example from quite some years ago now of like one of the main, findings that kind of blew up that world was that like if you could isolate CBD, it was shown in quite a large trial that it's actually quite useful for treating pain. So you guys probably are already aware that things like CBD are used to treat pain, and it's probably becoming more popular now. Right, so if you can believe it, that's all I'm going to tell you about the cannabinoid system. I just want to talk about some of the drugs that we can use to target different areas of that. So we can use drugs to either increase cannabinoid signaling, or we can use drugs to, say, downregulate the signaling. So if we're targeting CB1, we can give inhibitors to then obviously inhibit the receptor. So that would be an example of inhibiting the CB system. So there are drugs which can do that, and those drugs tend to target things like obesity, metabolism, and the craving effects that the ECS system has. We can also use CB2 agonists to, then upregulate CB2 function. So that's to do things like treat like pain and inflammation. Osteoporosis is also a very common thing, but it's not in the context of the nervous system, but it is a common thing that CB2 agonists are being used for. And then we can target the enzymes that actually break down the endocannabinoids themselves. So we can target an anandamide breakdown, and there's some evidence to show that if you inhibit the breakdown, we can treat things like pain. And it seems, at least at this stage, to have limited side effects. But I should caveat that all this research is still ongoing. We still don't know a lot about what these drugs and systems do. And then, like I mentioned before, we can actually target the transporter, right? So we could target the transporter, so we could inhibit the transport, and we might want to inhibit the transport to have the CB receptor, the cannabinoids around in the synapse longer. But it's quite difficult because many of these inhibitors actually interact with the CB receptors themselves, and also PHAR, which breaks down the enzyme. So this is quite a hot topic, but it's very hard to use it, because don't forget, you need transporters to move it from the postsynaptic neuron to the synapse, and then from the synapse into the presynaptic neuron. So if you inhibit it, you'd have to be able to, somehow not inhibit its transport from the postsynaptic neuron out. Otherwise, it's never going to make it to the synapse. So that's why it's quite a tricky area to work in. All right, cool. So psychedelics. So psychedelics, also a common psychoactive drug. I've shown here different classes, of psychedelics. Psychedelics are also known as serotonergic hallucinogens. And so this already tells you then that they work on the serotonin system. So some of these are quite common substances that you might have heard of in terms of psychedelics. So psilocybin, magic mushrooms. There are also things like certain species of frogs and toads in their skins that can cause psychedelic effects. LSD, mescaline. I've shown here in this paper, it shows things like MDMA and ketamine. These are sometimes considered psychedelics and sometimes not, depending on where you look at it, because MDMA and ketamine, they don't actually target the serotonin system, but they have very similar effects in that psychedelics are compounds that alter consciousness, right? And so while I put them here, these don't target the serotonin system, but they do alter consciousness, which is why some people talk about psychedelics. I personally don't have a personal preference. I don't care. Like, it changes consciousness. Okay, that's cool. Then we can study it. But just realize in some of your textbooks, like, you may not find things like MDMA and ketamine in there. They're very powerful psychoactive substances that they affect many cognitive processes, like perception and mood. They're actually considered to be some of the oldest pharmacological agents because there's a very long history of their use. So, if we go back many years, a lot of the Native American cultures used psychedelics for rituals and cultural purposes. So it goes back a long, long time. Things like peyote has been used for a very long time. Ignoring MDMA and ketamine, most of them are actually agonists or partial agonists for 5-HT receptors, right? So for the receptors for the serotonin system. These are the naturally occurring, LSD, and so the green ones here are naturally occurring, the ones in blue are actually synthetic. So LSD and MDMA and ketamine are obviously synthetic. So psychedelics can actually also be prodrugs in the sense that their parent compound that can be taken is actually inert, but the metabolite that they made is the psychoactive substance. So psilocybin is typically a prodrug because psilocybin itself is inert, but when it's dephosphorylated to psilocin, psilocin gives you the psychoactive effects. Now, psilocybin is mostly a prodrug, but if you give psilocybin intravenously, it can then have some psychoactive effects. But taking things like orally, psilocybin itself is a prodrug. And as I mentioned, some psychedelics can bind to non-serotonin receptors like MDMA, ketamine. So ketamine works on the NMDA receptor. So I think I've forgotten to say something. Oh, so with psychedelics, you get a wide range of effects, very individualized, right? So you've probably already heard anecdotal evidence. When some people take psychedelics, some people get great euphoria, they get some pretty wild hallucinations. But some people can actually have the opposite. They might have terrible experiences where they get like these fear hallucinations, right? So the variability of the effects of psychedelics is actually really, really wide. That's something I want to make clear. As opposed to something like cannabinoids, which is probably a bit more predictable, psychedelics is more unpredictable in their effects. So a little bit about LSD. So LSD was actually discovered by a chemist in Switzerland who was working at a pharmaceutical company. And from what I understand, this guy, Albert Hofmann, actually made synthesized a wide range of drugs. And it was only about like 5 years after that he made it is when he really discovered that it had some kind of psychoactive effects. Now, depending on what you read, he discovered it because he accidentally inhaled trace amounts of it, right? And that's where, that classic thing where the scientist does an N of 1 and finds something. It actually found it had these psychological effects. He then continued to kind of self-experiment and document his own experiences of LSD, right? So that's wouldn't really be done today in today's science. And LSD, has a very long history in terms that, it was made in the 1940s, originally for experimental testing. During the so-called hippie movement, LSD was really quite, prominent. Because of its uncontrolled use in 1970, 1973, Richard Nixon put this kind of restriction on it, which then kind of made it taboo and probably made it even more popular. Because of his discovery, actually in 1943 on April 19th, this is what is considered to be Bicycle Day. And Bicycle Day really is to celebrate the discovery of LSD, right? But I think that's more of an American thing. I don't think anyone celebrates Bicycle Day here in Australia. So a little bit more about LSD, or lysergic acid diethylamide. This is LSD here, This is serotonin here. So you can see they're very similar structures. So it's probably no surprise then that's why it kind of like binds to the same receptors. We know that LSD is targeting these receptors because very early work using selective antagonists was shown that the effects of LSD could be prevented if you blocked 5-HT2 receptors. And by the late 1980s, through the pharmacological work, it was known that LSD was working as an agonist or a partial agonist for 5-HT2A receptors. So 5-HT2A receptor activation activates signaling in transductive pathways in neurons through this G-alpha-Q signaling or through beta-arrestin pathways. And 5-HT2A receptors are really widely expressed in the brain, but in the cortex, they have really high expression in the dendrites of glutamatergic neurons. So glutamatergic neurons, as you hopefully remember, are your main excitatory neurons. Okay, they drive things like synaptic plasticity, just general brain function. And so then it's, if we have a high abundance of these receptors on these really important cells, then psychedelics can have that really powerful psychoactive effect on us. And because it's affecting things like excitatory neurons, it's affecting things like neural activity and plasticity. So LSD, it has this wide range of this strong psychoactive effects. What's, I guess, different between, say, LSD and cannabinoids is that LSD actually develops tolerance really quickly. So within about 24 hours after a single administration, you will develop tolerance. So, and the peak effect of LSD usually peaks within, tolerance develops very quickly within 24 hours and peaks within 4 days of taking it. And what's really strange about psychedelics in general is that they show this very strong cross-tolerance. So you can take one type of psychedelic and develop tolerance to a totally different type of psychedelic. And that's what I mean by cross-tolerance, that one psychedelic is developing tolerance to another psychedelic. So LSD will have tolerance to things like peyote as well. Why we get this tolerance is really a mystery. We still don't really know, but from what we do understand is that this tolerance to psychedelics develops through a downregulation of the density of this 5-HT2A receptors on our neurons, so like on our dendrites of glutamatergic neurons. And it also has a decreased binding affinity to these 5-HT2A receptors. So here I'm just showing a very simple table from quite an old study. And basically what it's showing you is the binding of, say, chronic administration of placebo or vehicle and the binding of chronic LSD and different brain regions. You don't have to know. Bottom line is, all you can see is if someone takes chronic LSD, they have a decreased binding efficiency or affinity of about 36%, right? So I don't expect you to remember any of the numbers or the brain regions, so it's just to illustrate my point. What we also know now through great gene technology is that if we knock out beta-arrestin signaling pathways of the 5-HT2A receptors, it doesn't prevent tolerance. Okay? So this tells us then that the impairment in the G-alpha-Q signaling or activation of G-alpha-Q signaling is probably the main driver of the development of tolerance to LSD. Does that make sense? Okay. And so, that's psychedelics and LSD. How can we then, harness the power of psychedelics as a treatment? So, like I mentioned, in 1970, there was a really a controlled ban on psychedelics, and actually put a real downer on the development of psychedelics in research as well, because it was actually very hard to use psychedelics once that kind of thing came across. It affected research as well. But we know that psychedelics, used in the right context, might be able to treat things like anxiety, depression, OCD, headaches, glaucoma. A lot of these probably overlap with the things that we use cannabinoids for. It just maybe provides us with a different way of targeting it. Cool. And so that's it, guys. I told you it would be a whirlwind, and hopefully it'll be easy for you to remember the key points of that lecture. So hopefully I'll see you Thursday for photopharmacology, I think it is.